Welcome to Wildlife Rehabilitation from Rescue to Release, a podcast dedicated to licensed wildlife rehabilitators and the veterinarians who support them. I'm your host, Gail Buell. Techniques we discuss here are meant for licensed wildlife professionals. If you aren't licensed and trained for wildlife rehabilitation, do not attempt to rehabilitate any wild animal. If you do find an injured wild animal, a wildlife professional can recommend next steps. We'll tackle topics about rescue, care, management, reconditioning, and releasing wildlife found in North America. This is a deep dive into each subject. We hope to give rehabilitators information they can use because they're already going to great lengths to rescue and nurse animals back to health. There are three possible outcomes in wildlife rehabilitation: release, euthanasia, and placement. An injured or orphaned wild animal's journey to restored health and independence is delicate and complex. We draw techniques and expertise from many disciplines. Many animals brought into rehabilitation cannot be saved. The last gift we can give them is humane euthanasia. We will talk more about when this is necessary. Sometimes animals are deemed non-releasable and evaluated to be suitable for a life in captivity. When that happens, we shift to a long-term approach to managing their care, training, and housing. These animals serve as ambassador animals, a term for the animals that rehabilitators use to educate the public. Each episode, we'll speak with experts who represent specialty topics across the profession of wildlife rehabilitation and ambassador animal care and management, showcasing best practices. So for this segment, I want to really concentrate on talking about a disease, a virus that has come to the United States called rabbit hemorrhagic disease Virus-2. We'll refer to it as RHDV-2 from now on because it's such a big mouthful. But Laura, could you talk a little bit about what this is?
Yes, so RHDV-2 is a calicivirus, which is in the family of viruses of like norovirus. It's a non-enveloped single-strand RNA, and why we care about that is that it lacks the fatty outer covering, so it's not susceptible to simple detergents like soap. So for those of us in the animal community, we can kind of think of this as being similar to parvovirus or the feline calicivirus in terms of hardiness and resistance. So this virus is extremely contagious. It's has very high rates of morbidity and mortality. And it's very, very stable in the environment. It can live over 100 days in dry conditions and can only take a few viral particles to induce disease in an animal. So it's a very important disease for us to be aware of because it's highly contagious and high rates of death in the rabbit populations.
This is very scary. When did it show up in the United States? It's been in other places around the world, right?
Yes, and it has a very long history. Basically, it was first identified in China in 1984, but it likely came from Europe. And this was the original RHDV virus, so not this subtype 2, but the original virus, again, identified in 1984, spread across Europe and Asia. Millions of rabbits died, escaped into other areas of the world. The original virus was first identified in 2000 in breeding colonies in Iowa, but that original virus only affected domestic rabbits. So there were not any big die-offs of wild rabbits because they're a different species altogether from domestic rabbits.
Okay.
So we, saw this spread and these, die-offs of domestics. In Europe, their wild populations are the same species as domestic. So there were wild rabbit die-offs in Europe, first in Iowa and domestic breeding colony, and then this new RHDV-2, so this subtype here, was first identified in France, again in a breeding facility. Again, kind of same thing with the first virus, that it was going around, there were massive die-offs spread throughout, the continent. And then this RHDV-2 came to North America, first identified in 2018 in British Columbia. And then in 2019, we started seeing some outbreaks in domestic populations. And then that kind of thing that's causing to get our attention now is that rapid spread and die-offs starting in, early 2020 in the Southwest United States.
In domestics or in wild rabbits?
In wild rabbits.
In wild rabbits.
So the RHDV-2 has mutated that can affect both wild and domestic rabbits, which is a very significant thing that, the previous virus only stayed in domestic rabbits, but this subtype is now contagious to and affects wild rabbits and hares.
Wow. So my guess is the listeners here all understand actually how important rabbits and hares are to the environment, but I'm going to ask this question anyway. Why should we care? There's rabbits everywhere.
Yeah. Well, as many of us understand, ecology, rabbits are at the bottom of the food chain. So when you think of, the stable ecosystems that our animals are part of, that we're a part of, if you take out the bottom rung of a ladder, you're going to have less stability. It's going to affect every animal and aspect down the chain. So we look at, there being an abundance of rabbits. It's not a species that we normally get concerned about, although there are some specific species of rabbits that are threatened and endangered. But there's many animals further up the food chain that are threatened and endangered. So if we wipe out their food source, makes it, that food scarcity, it's going to make it harder for these other animals to survive and thrive, raise their young. And we could see, potential decreases in populations of animals that maybe can't rebound as quickly, from that instability of the food chain.
And we've certainly seen over the years groups of animals that were very strong in population really go down and become endangered species, and actually in the past become extinct. Mostly because of anthropogenic or human-caused things that happened in the environment. But so that certainly, we're talking about eastern cottontail rabbits today, but Laura is definitely right. There's endangered and threatened species of rabbits in different parts of the United States and in North America that this virus affects. But, it can affect eastern cottontail rabbits too, and they're populous now, but this could really affect them. So we wanted to talk about this today because we think it's important because cottontail rabbits are the animal that comes into rehabilitation probably the most.
Yep,
unless you specialize in one animal that's not a cottontail rabbit, you're going to get cottontail rabbits, whether injured adults, but most often you're going to get young cottontail rabbits. And so this is important to all of us. Is this zoonotic? Can people get this?
No. So it is only affects rabbits. So it does not affect other species such as dogs or cats or, livestock hoofed animals. Mm-hmm. It is a rabbit disease. So, again, it can go between domestic and wild rabbits, but it is not going to infect and cause disease in humans or other species.
Okay. And you mentioned before that it is very contagious and it's very stable in the environment. How does that translate into a rehabilitator's facility, for example? Maybe we're jumping too far ahead here to talk a little bit about biosecurity, but that would be one of the things that I would be concerned about. I've got a batch of young cottontail rabbits and I'm getting another batch in. And how susceptible are young rabbits?
Yeah, so that's one of the things that with this new virus, we've been seeing that the young animals are just as susceptible as some of the older animals. They're maybe slightly protected in the environment because they're not going to be out foraging for food and potentially coming in with those contaminated particles as an adult mobile animal would be. Mm-hmm. But they have documented, disease, state, and death in young animals as well. So it's very important for us wildlife rehabilitators to think about. So kind of going back to the stability of the virus, here in Minnesota, it gets hot in the summer, it gets cold in the winter. I was really hoping and had my fingers crossed when I first learned about this, that it would, freezing would kill it. And, mm-hmm. At least if we had an outbreak, winter would wipe it clean. And they've determined that this virus can live, again, for greater than 100 days and can survive freezing and extreme temperatures. And so
it's like the evil villain in a superhero movie.
Yes, but us wildlife rehabilitators will be the superheroes and fight the good fight.
Ah, I
So
love it. Yes.
how we want to look at this for our facilities. So, we need to talk about biosecurity. And biosecurity is procedures intended to protect humans and animals against disease or harmful biologic agents. So we need to make that at the forefront of how we're interacting with the environment at large, but then specifically the environment that we control, which is our facilities, whether it's a professional wildlife rehabilitation facility or a home facility. So when we look at this, we need to kind of look at all of our procedures from, how we intake those animals to how we handle them and rehabilitate them, and also how we release them. So first and foremost, the most important thing is to not be bringing in animals. That's going to be the potential spreading of this virus, is bringing them, transporting animals. So not bringing in any animals to rehabilitative care that don't need to be. So we all practice orphan prevention at the kind of the pinnacle of what we do as wildlife rehabilitators. This adds in another layer of importance, that again, we're leaving the animals where they came from at all costs. So if that animal does not need rehabilitative care, to educate the public. And we can use RHDV-2 as another piece of our toolbox to, talk to the public into doing the right thing and leaving that nest of bunnies alone, monitoring for mother. The other thing is, once those animals do come into our care, is that we need to talk about, isolation. So the incubation period for this virus is 1 to 9 days. So any animal that's brought into our facility needs to be isolated and quarantined for 10 days, a minimum of 10 days, before we can kind of look at, kind of the second aspect of our rehabilitative care. And I don't want to say kind of a loosening of our biosecurity measures, but, that minimum 10 days is that kind of critical period to rule out disease process in those animals. So things we're going to be looking for in those 10 days are going to be signs and symptoms of the virus, or I should say, the active disease process.
Okay.
So if the animal is infected, oftentimes the first symptom we're going to see is death, sudden death.
Wow.
They might die without any outward symptoms.
Okay.
That can be hard to differentiate. In
a lot of cottontail rabbits have sudden death.
Yes.
Wow.
So that can be hard to differentiate. And this is where, working with your veterinarian and working with your, state wildlife health agencies, your Department of Natural Resources, again, the state wildlife veterinarian, and kind of discussing, reaching out to them as a wildlife rehabilitator and discussing when are the agencies and the experts, the veterinary professionals, when are they wanting to test? So
okay.
Working with those agencies to develop kind of a screening plan, because do you test every single rabbit that dies in your care? Do you only test the rabbits that die within those first days, those first 10 days of quarantine? So those are important things to consider and work with, again, those agencies and veterinary professionals for.
And they would be able to tell you too, like, okay, we want to test everything in that 10-day quarantine period. And here, this is how we want you to prep the deceased bunny is, we want, it refrigerated, is triple bagged, refrigerated, don't touch it, don't anything, or we're going to freeze it, or you're going to send it right away via FedEx, with dry ice. So they will tell you that kind of piece of information. So this is great. And rehabilitators are often on the front line of being able to supply data, in this case, young dead rabbits, that may help inform what we might be able to do about this disease. So anyway, I'm all about data collection.
Yes, correct. So, and of note, they specifically do not want cadavers to be frozen. So while you work on getting, a potential specimen, to the agency or to the lab for testing, refrigeration. So just again, we'll talk about, the security of keeping the cadaver, the body,
Sure.
But yes, freezing does render it unable to test. So,
okay. Okay.
So again, those outward symptoms that we're going to be looking for, death obviously is the number one. Other things you're going to be potentially seeing, it's a hemorrhagic disease. So it's bleeding disease. So you're going to be looking for signs of bleeding in an animal. So you might see blood coming from the nose, mouth, or rectum. You can see respiratory distress in the animal. Lethargy. So if this, otherwise healthy animal is all of a sudden just very lethargic. Looking for jaundice, so that's yellowing of the skin or conjunctiva. If you listen to the rabbit's heart, you may pick up a heart murmur as that cardiac tissue is damaged. We're not very routinely necessarily taking, temperatures on our rabbits, but they might also have a fever, if you are assessing that as kind of biological data for your admission. So if you, again, are doing that, you might. And the reason we're seeing those symptoms is that the virus attacks, the liver tissue. And so that's why we're seeing those bleeding disorders. And that bleeding is because it's, affecting the liver, which helps with clotting factors, also affects the spleen, which again is involved in clotting and other areas. Attacks, you can get bronchopneumonia. And so therefore you can get kind of lung hemorrhage and edema, so swelling, in the lungs as well. So that's why you see the bleeding and the respiratory distress.
Okay.
But obviously those are all very heavy areas of damage to it. So that's why death might occur before you see those external symptoms, because you need your lungs, you need your heart, you need your liver.
Wow. How, before, I know we're going to really get into biosecurity here, and this I think will kind of lend itself to that. How do rabbits spread it? I mean, how does it get spread? Or how does this virus move? Maybe I should say that. Maybe that's more accurate.
Yeah, so there's a couple of different ways. So there's direct contact. So obviously, if animals are coming in contact with, infected feces, urine, blood, respiratory secretion. So if there's any direct contact between animals where one's infected, the other one is likely going to pick up the virus as well. Also, because it's such a stable virus in the environment, excuse me, we also see indirect spread. So contact with an infected surface. So in rehab care, that's going to be our cages, our exam tables, our feeding supplies. But out in the environment, that's going to be potentially shared water sources. If there's multiple rabbits that are coming to eat bird seed under a bird feeder, that kind of shared food source. And then the frightening part about this is that there's kind of also mechanical vectors. So we're seeing that, predators and scavengers and potentially insects can spread the virus. It can help it move greater distances.
Wow.
So we know our rabbits aren't, migrating. Our rabbits aren't flying from one state to another.
Right.
But birds do, and birds that eat rabbits do. And so we can see that shedding from the virus, from the movement of other animals, and that also includes people. So as this environment is as this virus is, in the environment, you take a hike, there's been, an outbreak, there's potential viral particles all over, the national park you walked through because there was a massive outbreak in the Southwest United States. And then as you fly home, if you don't properly disinfect your shoes and you go walking in your backyard, walking in your house, walking into your rehab facility, potentially you could be then spreading that virus as well.
That is incredibly scary. I've said that a couple times already, but it's incredibly scary because you make one slip up, you forget, you don't hear the news, you don't whatever, and you've just spread this virus.
And it is scary, but that's also why, knowledge is power. So we know where this is in the United States, and we're sharing that information now, via your podcast. There's great Facebook groups for wildlife rehabilitators that are constantly updating and sharing the information. State agencies are usually putting out, memos and press releases. But that knowledge is power. So as here in snowy December, Minnesota, as we're recording this, a lot of us are maybe taking a vacation to a warmer climate. Well, that Southwest United States is where the virus, has had an outbreak. So having that knowledge of saying, okay, if I go to a state that it is now considered, kind of a stable endemic area. It is, the environment is saturated potentially with this, this virus. If I travel there as a human, it is my responsibility as I come back home to, make sure that I'm disinfecting and washing my shoes and my clothes before I continue to wear them. Right. So, using that knowledge to be responsible travelers as humans is going to be one thing that we do have in our control to prevent the spread of this from the areas in the United States where it's now endemic to new areas, which is what they're continuing to see, is that, kind of the trickle spread of this across the United States.
Right. And I was going to ask you if it's now endemic, and it is. I mean, it's not everywhere in the United States, but it eventually will be wherever rabbits are.
So yeah, it's the USDA has considered it endemic in 11 states, and there's cases in both wild populations of rabbits and hares and domestics. And then there's 6 other states in the United States that have had kind of isolated domestic cases, including here in Minnesota. We had, for what at this time remains, kind of an isolated case in some domestic rabbits.
Okay.
So we'll have to kind of just continue to be vigilant and to, be as responsible as we can to try to make sure that it stays isolated and there's not outbreaks.
Right. Okay. I'm an in-home rehabilitator, or maybe a center. And I know that I'm going to have multiple batches of cottontail rabbits, and we'll start with babies because our focus, most people get a lot of young animals. So what do I do?
Yep. So you're right. As we digressed and went off on that left turn. So we started talking about quarantining. So that minimum 10-day quarantine when these animals come in. So as you develop your biosecurity plan for your rehabilitation, you're going to have to determine, whether you're going to quarantine every individual or every batch. Obviously you don't need to separate babies of a litter. They are kind of one entity. They're kind of one biosecure unit, if you will. But if you get in a singleton, you'll have to work up a plan to determine if you're going to keep that one isolated and separate from other ones, or if you're going to look at your quarantine and maybe, group your individuals to make maintaining, a biosecure facility feasible for you. Many of us have limited resources, again, whether it's time, space, financial resources, cage space, et cetera. So we need to kind of develop a plan that's going to ensure that we're compliant with this biosecurity plan. So again, looking at that quarantine, is it each group, or is it going to be, a whole day's admits? Because that's going to make working with those animals and caring for those animals maybe a little bit less time consuming. So it's something you'll work into your plan. With that quarantine, you're going to have to make sure that you're using, personal protective equipment, PPE, because you can't go and handle one unit of rabbits, whether it's a litter or a day's intakes, and then immediately go, even if you just wash your hands, go and handle another one. So that's points of cross-contamination. So looking at that, so you get in your litter of bunnies, you've got your, personal protective equipment, your gloves, your gown, your supplies, those are going to stay with that group. And make sure that you're maintaining the isolation of those animals and not cross-contaminating between your cares or your facility. So with that quarantine, again, you'll have to make sure your cages are a minimum of 3 to 4 feet apart. So that's where, again, looking at how you're going to do that feasibly in the space you have. Are you going to have an entire shelf that's an isolation unit? So the whole thing top to bottom is one unit, and then the shelf that's 4 feet away is a whole different unit. Would you cover,
I'm almost thinking not really greenhouse, but, but at a doctor's office where they have the curtains and stuff, would you, and maybe we'll get into what it looks like, but I'm picturing kind of a whole shelving unit and covered around with plastic or something to really keep it separate from another group of rabbits.
Yes, but also within reason and using your common sense. So to make sure that that our rabbits have ventilation and airflow and fresh air to breathe. So we can't put them in a plastic bag and seal it.
Good point. Good point.
Yes. However, yes, it's spread, again, that direct contact and that indirect contact. So it's kind of, if you think of droplet contamination. So it's not airborne. So the animals in a facility or room breathing the same air isn't going to contaminate them. But if the rabbit has, gets respiratory distress because its lungs are swelling or hemorrhaging and it's sneezing blood particles, you need to want to try to contain those bunny sneezes. So having, yes, plastic barriers, or, again, depending on your facility and the space you're using, you could even look into setting up like a tent and having them housed inside the tent as your quarantine area. Enter and exit. So you can get creative with how you do this in your facilities, depending on the space you have. But yes, a minimum of 3 to 4 feet between cages. And I absolutely, would advocate for some type of physical barrier between them to prevent, again, the potential spread of kind of droplets or, again, things falling out of cages. So you don't want, particulate matter. So like hay or feces from one cage, you wouldn't want to shake out a towel and have it, spread that around the room.
Sure.
These barriers also help serve as visual cues for us as caregivers. So we don't break that isolation and we don't cross-contaminate.
Okay. Well, maybe so each quarantine unit would have its own dishes. It would have its own hay or whatever kind of material you're using inside the cages. You'd already have a set of disinfecting tools and extra cages if you need to switch the bunnies to a clean cage. When you brought formula in, it just is for that group. Garbage is its own little place, and then you have to figure out how to take it out, but we'll talk about that, I'm sure. So am I kind of getting the idea of what this looks like?
Correct. So each cage or each isolation unit is going to need basically its own little self-contained wildlife rehab setup. So as you said, feeding supplies, the actual food or feed, you can't go around with a big, bag of hay and go from one cage to the next. So as you're doing your cares, you'll have to kind of make sure that you enter the isolation zone, put on your PPE, enter the zone that you have all of your supplies and duplicated for each unit. So you're not using, you're not washing those syringes and then going to another unit. Nope, those are going to be washed and reused for the same unit and the same, litter of rabbits. So yes, everything will kind of be duplicated within each area. Okay.
And that makes sense. Now, as far as your PPE, personal protective equipment, it's actually protecting the other rabbits, right?
Correct.
You put, how does that work? You put it on before you go in, you have it on the inside and you walk in and you put it on like the lab coat or whatever else. I mean, what does that look like?
Yeah, so we'll include this in the resources because there's really great information on how to put on and how to take off.
Excellent.
PPE.
Okay.
It's a kind of a visual process, so I can't really explain it very well over a podcast. But you'll have essentially kind of a transition zone before you enter any of these quarantine areas. I like to kind of think of them as the red zone, the yellow zone, and the green zone. So our red zone is our potential contaminated area. That's our quarantine or isolation area. Green zone is, green is good. It's in the clear. These animals are healthy. They've passed their isolation, minimum of 10 days. They're now, in the green, the healthy side. And then the yellow zone are kind of these transition zones where there's potential for, contamination. And that's kind of where you need to be extra thoughtful in your process. So I strongly encourage people to use visual cues. Again, we talked about those, the plastic barriers, but also colored tape. So, put a strip of red tape on the floor. Okay, once I enter the red zone, I cannot exit it without appropriately removing my PPE and, disinfecting. So you're going to put on your PPE, your gown, your gloves, your shoe covers before you enter it. So you come from green into yellow, you put on your PPE and then you enter. And that yellow zone can be kind of a staging area where, while you're still, not in the red, you can make sure you do your triple checks to make sure you have all your supplies and everything you need. So when you enter that zone, you don't have to then step out and potentially cross-contaminate.
Right.
The fewer transitions in and out, the safer, because any transition is, a potential route for cross-contamination. So you want to be thoughtful in your process and make sure your plan. So kind of triple check everything before you enter that zone. So yes, you'll put on your PPE before you enter, gather all your supplies, put on your PPE, enter your red zone, do the care of the animals in that isolation unit. So again, that's whether that's going to be, each litter of rabbits or a day's intake or a shelving unit, however you have it set up, that's feasible and realistic and sustainable for you. Do all your cares. Again, kind of talking about when you mentioned the garbage, we'll kind of talk about then, as you're doing your cares, you're doing your feeding, things like that, you need to make sure that, again, you're not shaking out that towel and sending those particles flying. So a big consideration for this is what materials we're using in our wildlife rehab. So we all like to reuse, towels and to clean them. We're all probably pretty frugal as wildlife rehabilitators. But this, it's going to be something that we're going to have to look at investing in safety and biosecurity for our rehab, and maybe look at using more disposable products. So we're not trying to exit with contaminated towel materials to then try to wash and disinfect them.
Right.
So, looking at using, puppy pee pads, or, some other thing like that is going to be disposable, again, compared to having to relocate and disinfect other material.
Right.
Along those same lines, another thing that is a challenging aspect of wildlife rehab is we really want to improve the welfare of these animals and provide them with natural material for hiding and digging in and food sources. But if you have RHDV in your area, we know that it's stable in the environment. So bringing outside material into your wildlife rehab facility could be potentially bringing in viral particles. So bringing in, a branch of a tree with leaves on it for kind of a natural hiding area to kind of, mimic, brush material, or foraging wild foods. So they have, the type of food they would eat once they're released, bringing those materials in. We can't soak grass and plantains and things like that in bleach before we give it to our rabbits.
Yeah, kind of defeats the purpose. Yeah,
so,
a little bit.
We might have to look at adjusting, some other aspects of our care for these animals to ensure safety and biosecurity. So again, not bringing in outside materials. So we'll have to get creative in how we're improving their welfare and their cage setups, without cross-contaminating anything else.
Okay. You mentioned a couple times disinfecting when you're coming out of your quarantine and disinfecting. And you mentioned at the very beginning that this doesn't get affected by soap because it doesn't have that fatty layer on the viral particle. So you mentioned bleach. What disinfectants can be used?
Yeah, so there's a whole list of them provided by the EPA. Again, I'll make sure that that's included in the list. The big ones that we're probably familiar with and use in wildlife rehabilitation is going to be bleach, Virkon, and Rescue. The important thing with any type of disinfectant product is to follow the instructions. So making sure that you're diluting or reconstituting it per the manufacturer's instructions, and then contact time. Bleach, takes, oftentimes minimum of 10 minutes with a good saturated contact time to disinfect that surface. The other important aspect of disinfecting is that it has to be clean first. So if there is organic material on a surface, so if there is, smeared poop on the cage, you have to wash and remove that organic material first before disinfecting the clean surface. So we often think of cleaning and disinfecting as one step, but really it's a two-step process, or maybe even three. First, you have to remove the organic material, wash the surface to get any of those other particles off, and then disinfect it.
Right. So you can mechanically remove some of these particles. And then, of course, then you're disinfecting to try to get the rest of them as best you can. And that's the thing is we need to read the instructions. I mean, all of us are in a hurry. All of us have so many things on our mind to do, but this particular, and I've been guilty of this, not having enough contact time, or like, oh, was it 10 minutes? I don't know. I wasn't keeping track. So we need to be a little bit more diligent. And I'm sure a lot of our listeners are very diligent. I have to be more diligent. There we go. I've said it. And, the other thing I was wondering about, especially with bleach, you can usually make it up into dilute it with water in a big container, but that only lasts so long before it is, I don't know, call it denatured or doesn't work anymore.
Correct. Yep. Yeah. Bleach, once it's diluted, is really, it starts to lose efficacy after 24 hours. Yep. So you need to be, mixing that up daily to be using for your disinfectants. The other products, again, you'll have to look specifically at their the manufacturer's guidance or instructions, but making sure that you're paying attention to all aspects of that is critically important, so that we're, again, being effective in our biosecurity measures for our animals and our rehab facilities.
Okay. So what else? I'm scared to ask. What else?
So we're going to, isolate and quarantine for 10 days. That's, again, we're watching for those signs and symptoms. We're working with our veterinarian and our state wildlife officials regarding when we're going to test or not. We're taking care of the animals, each group, everything one and of itself. So this whole group has everything. When we exit those red zones, we need to be mindful, again, because that exiting is when we can be potentially pulling those particles out with us. So removing our PPE carefully, so we're not, again, shaking that out. Having a plan for that regarding, are we using disposable isolation gowns? Are we using, say, lab coats that we're putting on backwards so we're fully covered in the front? And do we have a hamper, specifically kind of designated there, kind of a biosecure hamper, with, again, that's not permeable. So a plastic bag that we can carefully put that in until those can be appropriately laundered and disinfected. How are we going to get our trash out of these areas? So we talked about having a garbage for each little area. So when we need to remove that, not all of our facilities have a garbage chute straight to the outside. Many of our facilities are interior rooms. So we might have to walk through other spaces that are going to be clean, and we don't want to cross-contaminate. So looking and having a plan to, say, taking your garbage bag out, having, again, this kind of yellow zone where maybe you take your garbage bag out of the red zone and put it in a Tupperware bin in the yellow, spraying the outside of that bag off with a nice good bleach solution, letting it sit for 10 minutes, and then that bag can be removed, through the rest of our facility, or, green zones, out to the village, however that is.
Okay. Yeah. So extra steps. I mean, we're all very diligent about making sure things are clean already and everything is good for the animal. This is just definitely next level up.
Yep. Yeah. It's extra steps and extra time. And I think that's one of the most important considerations as each individual rehabilitator facility develops their biosecurity plan, is looking at what is realistic for them. So we might have to look at some hard realities of what we're going to admit to our facilities.
Right.
Some areas the choice may be made for us by, basically geographical bans, perhaps, if it's known to be in an area, your governing agencies may say you cannot accept animals from these counties.
Okay.
If the virus has been identified there, our governing agencies that permit us to be wildlife rehabilitators, could potentially just put a ban on the rehabilitation of species altogether.
Right.
I would advocate that we should work with them and talk to them, show them how prepared we are, show them how professional we are. So hopefully we can still continue to help and support the animals that need us. And ultimately too, if there is, massive die-offs in the wild, that perhaps our activities can contribute to the restabilization of that ecosystem and food chain. So having those
Yeah.
discussions and working with those agencies to prove that we can still do wildlife rehabilitation safely of rabbit and hare species with considerations for RHDV-2.
Right. And it is being proactive, and it's showing that we're partners and that we're not just for, and I know we're not as a group just for the individual animal, even though that's what we're caring for, that we're really looking at the population and population level care as well. Because I don't think any of us want to help spread this disease. And we have been working with other diseases too, right, in different areas, whether it's chronic wasting disease in white-tailed deer or deer species, or things like white nose syndrome in bats. Well, a lot of what you've talked about is very similar. I do a lot of work with bats, and it's a very similar challenge with PPE and making sure that you're clean and not spreading it. That particular virus, we have figured out how we can work with that virus and kind of kill it, and still work with the bats. Chronic wasting disease is a completely different animal, but white nose syndrome, we are, and that's been in partnership with the regulatory agencies. It's been in partnership with scientists who are desperately and had been desperately working on trying to figure out that disease so we can save bats in North America. So some of this hopefully is slightly familiar, although that this disease still, to me, I'm saying it again, it's very scary. Okay, so we have, let's say, an isolation area with a set of rabbits, and now I'm getting another set of rabbits, or maybe a singleton, and maybe it's a week into my quarantine before my group A. I get this singleton in, kind of, is there, how we always want to say, okay, well, I've got to examine this one, and I've got to, or this batch, and I have to give fluids and look at this stuff. Oh, but I have a feeding for the other batch at noon. I mean, is there an order to how I have to look at these animals when they come in?
Yeah, so we want to go from the safest animal to the least safe in terms of biosecurity. So the safest animals are going to be the rabbits in the green zone. They're out of their quarantine. So you should be starting your chores, starting your cares with those animals that are the most established in your nursery or facility, which is opposite of how we normally do it. We want to go stabilize those potentially critical admits first. Before we go to the ones that are healthy and doing well. But we need to be very mindful that, again, we're not cross-contaminating. So while you're in your areas, again, you're going to do all your chores for those rabbits in the green zone first. And even in the green zone, you should really, try to just be mindful. And also, if you're mindful in how you're doing this in the green zone, it'll transfer through to the red zone as well. So just kind of keeping it the same is going to help you be compliant with your plan. So starting with the most established rabbit in your care and working backwards. Okay. And then kind of doing that again in your red zone as well. So you're going to be taking care of the rabbits that are closest to the end of their quarantine period, that minimum of 10 days. Taking care of those first, and then working back to that newest rabbit, that newest admit, along the same kind of thought process of taking at admissions, is really kind of, we're talking about, setting realistic goals for the intake of these animals. Again, they might be geographical limitations, but it might also be time frame. So if you and other rehabilitators in your area typically collaborate and transfer animals to and from each other, you're going to have to stop that, and you're going to have to plan accordingly. So say you get a phone call on this singleton rabbit and you're at capacity, but another local rehabilitator has space, but they can't take the rabbit until, say, tomorrow afternoon. Historically speaking, I may admit that rabbit, provide stabilization to it, and then work to transfer it to that individual. But that is, too many points of cross-contamination. So in these things, we need to be thoughtful about, talking to the public and getting that animal admitted to the kind of the end facility, the facility that's going to provide the definitive care for that animal. Reducing the transport and transfer of these animals. So we might have to kind of go against our desires and maybe give the public some kind of emergency or overnight care instructions to help keep these animals stable, or, as stable as they can be until it can get to the definitive site for rehabilitative care.
Sure. I also see there's an opportunity here, right? I mean, we like to collaborate with each other. We do transfer animals back and forth, but there may be an opportunity here that says, hey, Gail, you get the first week of the month admits, and Laura, you get the second week. I mean, it's one possible way of working together, right? Yeah. And then Ellie gets the third week and somebody else gets the fourth week, and then we start again, or whatever, we can do it in 10-day increments. So, but yes, I think organizing in your area is going to be really important. Of course, this is also a call for more people wanting to do wildlife rehabilitation, because we are always in need of more skilled hands for whatever in rehabilitation, not just rabbits, but in lots of different species.
Correct. Yeah. But if your own facility has limitations in space that you can't set up multiple zones for rehabilitation. So in a perfect world, you have completely separate rooms. You have a red room and a green room, and you have completely separate rooms for those animals in your biosecure facility. Many of us that are home-based wildlife rehabilitators don't have several rooms at our disposal. We have one, maybe two. And so if partitioning your room, into sides and using, those barriers, so hanging a tarp down the middle of your room to, differentiate the green side from the red side, if that's not possible or practical in your facility, exactly, collaborating and developing a biosecurity plan with several rehabilitators is a great way to get creative to still serve the need. And be able to have that isolation and quarantine of animals. The challenge in that you'll have to discuss, and even with your own home facilities, you're going to need to have kind of contingency plans. So if I take everything for 2 weeks, minimum of 10 days, so let's just break it up into nice 2-week periods that's, consumable to the mind. I may get overwhelmed in that 2 weeks. As we know, rabbits kind of tend to go on a cycle altogether, and I'll get a whole bunch of phone calls and a whole bunch of admits, and then I might not, get many over the next couple of weeks. So how does your group of rehabilitators that has this plan, how are you going to handle if a rehabilitator gets, hits capacity for their facility or their time allowances for care in the number of animals coming into your area? Triage and, potentially and very likely euthanasia is going to be something that you'll have to discuss and have a plan for, because each step of care is going to take a lot longer, because you have to, again, have separate things, you're entering and exiting, and all of our care of these animals is going to take more time than it historically has. So looking at the realities of what you can provide quality care to these animals to, and be safe and biosecure in how you're doing it, you're going to have to discuss potentially kind of intake parameters for the animals coming into your facility, working with your group, working with your veterinarian to determine, kind of what's, acceptable to you as a rehabilitator and a caregiver of these animals.
Right. Yeah, a lot of things to discuss. There's also, just to remind everybody to throw this in there, is that the code of ethics from NWRA and IWRC, also the, like in Minnesota, we have our study guide, but there's the standards of wildlife rehabilitation at the national and international level. All of those things can kind of help guide you a little bit with what you're comfortable with and in doing rehabilitation and helping you make some decisions. What else should we be thinking about or talking about with RHDV-2?
Again, if you have a suspected case, we've talked about working with your veterinarians and testing, but as far as treatment goes and the ethics of working with a potentially diseased rabbit.
Can these rabbits recover?
Some do. Looking at what they saw in the Southwest United States over the last year, the mortality was about 90%.
Okay.
So some rabbits are surviving this virus and this disease process. There's data showing that they can, shed viral particles for 2 months.
Oh my goodness.
So if they're an asymptomatic carrier, if they survive the virus, they're potentially spreading this. So this is something, again, you'll need to work into your plan about, maybe holding any animal for, 2 months after its quarantine period is cleared before releasing it. We know rabbits are a high stress species. We like to release them the minute they're ready, but is that best practice to make sure that we're protecting the greater environment and our ecosystems? Looking at making sure we're releasing animals back to the geographical location that they came from, if they are healthy, so we're not spreading potentially from one area to another or from our facility to another, which should not happen if we're careful and practicing our biosecure plan. So if an animal does get, again, just have signs and symptoms, there's not a cure for it. We don't have at this time a viral drug, an antiviral drug we can give it to stop the disease process. It's basically supportive care, but you need to really consider, and again, with the guidance of your veterinarian or your partners, to look to see, is it safe and is it ethical to treat that rabbit in trying to prevent the spread of this disease, again, to other rabbits in your facility or the greater, again, ecosystem or environment if that rabbit does survive and get released.
That brought up another question. Is there a vaccine?
There is a vaccine approved for use in domestics.
Okay.
Right now, it is not approved via the governing agencies for widespread use. They're authorizing it for use in each state that cases are identified. So if your state has confirmed cases of RHDV-2, then your veterinarians can look into importing the vaccine from overseas and administering it to domestic rabbits. There was in California, they did do an experimental vaccination of some endangered brush rabbits.
Oh.
And so they were trapping these rabbits, they were vaccinating them, they were monitoring them to make sure that there weren't any, harmful side effects or, unintended effects from vaccinating these wild animals with a domestic vaccine. And then they were ultimately releasing them back to the environment. So that's exciting news for us wildlife rehabilitators that there has been use of vaccines in, again, a wild rabbit species here in the United States. At this time, again, it was isolated to that endangered species. But the work that they've done on that is going to set the foundation potentially for, future use and future vaccination of, wild rabbits in the United States.
Wow, that gives me hope. That gives me, because it's kind of dire, but that just, oh my gosh, science is amazing. Science is amazing. And yeah, that I'm glad that people were thinking that far ahead, going, hey, can we try this out on this endangered species and see if it doesn't work? So yeah, that does give me a lot of hope.
As you know, I work in human healthcare. So a lot of this kind of transfers from my one area of work to my wildlife rehabilitation volunteer work. But just having somebody that can go through your plan with you and watch you as you practice it. But before you get into the busy wildlife rehab season, you've got rabbits coming out your ears, you're completely overwhelmed, and then your governing agency announces that there's been a confirmed case in your area. You don't want to have to try to pivot mid-season when we're burnt out and overwhelmed. And try to, redo your whole workflow. So setting it up in advance, having somebody watch you, practicing it, and then, realize that you're going to probably have to pivot and adjust things as you go through the season. Be nice to yourself. Tap yourself on the shoulder for doing a good job on this, that, it is overwhelming. It can get stressful, but taking that deep breath and reminding ourselves why we're doing this. And that even though it's scary and overwhelming, that, there's animals at the end of the day that need our care and assistance, so they can resume that wildlife. So be nice to yourself. Don't get discouraged. And then just get creative with it. As we talked about, you could set up a tent, a camping tent in your room, tarps down the side of it. Instead of using open-sided cages, using Tupperware bins and putting, kind of a mesh top on it. So any, hay or stool, other contaminants are kind of contained in that cage versus maybe, historical caging that we've used. So being creative, we're a resourceful group. We're kind of creative, and
all the time.
People donate us stuff, and you look at it and go, I will find a use for that. That's how we're going to have to approach, biosecurity in our home facilities when we don't have unlimited resources to invest in this. So get creative, work in a group, again, practice it, and just know that what you start doing may not be the only way it can be done. Again, there's principles and best practices that have to be followed to be safe and secure, but knowing that you can make adjustments and change things up throughout the season, as long as you're adhering to those biosecurity principles of not cross-contaminating, how you enter and exit zones, keeping those rabbits, isolated from each other and quarantined.
Excellent. We will have resources on our webpage for the podcast. And one of those will be the IWRC, International Wildlife Rehabilitation Council organization. They come together with at least a general protocol for RHDV-2. And Laura, you have graciously said that you will be putting some resources together for everybody to look at and follow, including links to videos to show how to don and doff and all that, your PPE properly. Laura, I want to thank you so much. This has been so informative and such an important topic, and I'm hoping that I can invite you back again as we learn more about this particular disease and as we get updates. So hopefully I'll have you back again. So thank you.
I look forward to it.
Good. Excellent. Well, thank you so much again. Much, much appreciated on this incredibly important topic. This was part one of my talk with Laura Davich Jette, one of my go-to experts on eastern cotton-tailed rabbits. Now that we've covered RHDV-2, we dive into the details of caring for orphaned eastern cotton-tailed rabbits in episode 14. Please join us. This was another episode of Wildlife Rehabilitation from Rescue to Release. We're glad you joined us today. Wildlife Rehabilitation from Rescue to Release is a production of Partners for Wildlife, working hand in hand with wildlife rehabilitators and veterinarians to improve the welfare of orphaned, ill, and injured wildlife. Our executive producer is me, Gail Buell. I'm the Partnership Coordinator for Partners for Wildlife at the University of Minnesota College of Veterinary Medicine's Raptor Center. To learn more about the Raptor Center and Partners for Wildlife, visit Raptor at umn.edu. Our producer is Kirk Clockey. Are you interested in becoming a licensed wildlife rehabilitator? Visit the National Wildlife Rehabilitators Association website at nwrawildlife.org for resources to get started. And remember, if you do find an injured or orphaned animal, please contact a licensed wildlife rehabilitator in your area. And that's our show. Hope you'll join us next time.